2021. Just to briefly introduce myself, my name is Rachel Leanne, and I'm this year's TMD co-chair. And just a quick disclaimer before we get started that we are recording this meeting, so you're free to turn off your camera and mute yourself and just chat and just utilize the chat function. But we do encourage you to turn on your camera if you want to and interact with our wonderful presenters. All right, so we'll go ahead and get started. I wanted to give a little bit of an introduction on what is a startup. So startups generally refer to companies that are in the early stages of development. They're typically founded by 1 to 3 founders who aim to turn a vision into a successful business. And in this case, in translational medicine, startups usually aim to offer innovative medical devices and treatment technologies to the public. So some interesting quotes that I found of founders of startups when they're asked what a startup is, these are some things that they said. So one quote is, "A startup is a human institution designed to create a new product or service under conditions of extreme uncertainty." And another quotation that I found that I thought was interesting was, "A startup is the living embodiment of a founder's dream. It represents the journey from concept to reality and is one of the few times when you can take something that is only a dream and make it a reality, not just for yourself, but for the entire world." And so those kind of embody the spirit of startups. And I think there's something really special about being in this space of uncertainty and excitement. And that's what we're hoping to kind of capture in the session today. So startups in reality are multifaceted, diverse, and hard to define. These are just some of the characteristics that might define a startup. So focused on growth, solving a problem, a lot of uncertainty, filling a gap in the market, changing the way things are traditionally done, not successful yet, only for the ambitious, a few employees, adaptive, and independent. So I think we'll see from our presenters how many of these things they've experienced and what working on a startup has been like for them. And so I'm really excited to introduce our wonderful set of presenters today. So starting off with Impedia Tech, we have Rafaela Samoas joining us, and she is an undergraduate researcher. We also have Dr. Debbie Chen, who is the founder and CEO of Hydrostasis. And we have Greg Poor, the Chief Analytic Officer of Micronova, as well as Dr. Joseph Carson from Pensive Vision, who is their Chief Technology Officer. And so the format of the session will go in the order that you see here, and each startup will present for about 10 minutes. And then after each startup has presented, we'll have a Q&A session at the end for the remaining about 10 minutes of the hour. All right, so without further ado, I'm going to turn it over to Rafaela.
All right, thank you, Rachel. Let me just share screen. All right. Can everyone see my slides?
Yes, we can see it.
All right, thank you. And hi, everyone. Thank you for having me here. Some of you may already know me. My name is Rafaela, and I'm here to talk a little bit about translating academic research into market and how our team in Professor Talka's lab is doing that for Impedia Tech. So our project was inspired by the motivation to solve a problem, which is the incidence of catheter-associated urinary tract infections, which are one of the most common infections acquired in a hospital setting. And it's expected that about 15 to 25% of those patients who are hospitalized will have to use a urinary catheter during treatment. And this comes down to an estimated 160,000 cases in the US alone each year. And the Centers for Medicare and Medicaid Services consider CAUTI a never event, which means that hospitals are responsible for the full cost of treatment if it is acquired during a hospital stay. So it makes this cost of treatment about $13,000 per patient. And we know also that prolonged catheterization poses a large risk for developing CAUTI, so having a catheter placed in for a very long time. So why is that? And we're looking at bacteria, which exists in two different forms, as either planktonic or free-flowing bacteria, or as biofilm. And planktonic bacteria is a little bit easier to inhibit, but the big concern for the management of cavity is the formation of biofilm on the surface of urinary catheters. And biofilm is composed of an agglomeration of cells that kind of stick together and form an extracellular matrix, which makes it less responsive to antibiotic treatment since drugs aren't able to diffuse as easily through the extracellular matrix that the biofilm forms. So our solution to this problem was to develop a catheter coating that can be applied to the surface of a urinary catheter, and it uses various mechanisms to inhibit bacterial proliferation. And our unique formulation of active materials includes a mixture of heavy metals and PDMS, which is a medical grade silicone, and that allows for bacterial inhibition through mechanisms that include the formation of electric fields, hydrogen peroxide, silver ion toxicity to the bacterial cells, and for the formation of barriers that prevent cell attachment to the surface. And when comparing our technology to that is commercially available, we see that we're able to approach the bacterial inhibition through multiple methods, which makes our coating superior to commercially available coated catheters. And again, we should highlight that we don't only rely on heavy metal ion toxicity, which is a fairly common technology used in urinary catheters, but we also use additional mechanisms that can be effective at inhibiting bacteria. And we're able to approach it on the surface of the urinary catheters. So to validate this, we conducted multiple experimental tests, and not only were we able to significantly inhibit planktonic bacterial growth for the entire duration of our experimental setup, which is 6 days, but we were also able to inhibit biofilm on the surface of the urinary catheters. And comparing it to existing coated catheter technology, we proved that our coating is effective at both inhibiting biofilm and at inhibiting planktonic bacteria for a longer period of time. And UCSD has a lot of resources to help students and researchers translate their technology, and some steps that we've been taking to carry our technology forward was to file a patent, which we did it through the UCSD Office of Innovation and Commercialization. And we've been active in the academic community, having published 2 journal papers and recently submitted our 3rd conference paper. And when seeking funding, we relied mostly on academic grants, and we're now funded by the AIM grant, which is a program designed to help technologies developed at UCSD be translated into market. We've also been advancing our business efforts by participating in both the UC Biomedical Pitch Day and in the MedTech Accelerator program, where we even further dove into our commercialization efforts and received feedback from business experts on how to plan our marketing strategy, which for this technology is to enter the market as a premium product for patients in the ICU at first, and then expand our target market once we're established in this area. We're also working on continuing research to further characterize our technology and to optimize the amount of active material in our coating so we can lower the price of our product while still maintaining the level of effectiveness that we've gotten. So here you can see the timeline of our project, and we started in 2018, but currently we're looking at setting up clinical trials to judge the safety and clinical efficacy of our coating, as well as continuing the academic research side in parallel to the development of our company. And a really big part of being able to develop this coding relied on having a great interdisciplinary team consisting of medical professionals, biologists, and engineers. And with this, we were able to get together and analyze the problem through multiple perspectives to come up with this solution. So in summary, we know that CAUTI is a huge problem, and it's one of the most common hospital-acquired infections. And to approach this, we developed a coating that was able to approach bacterial inhibition through multiple modes of action. And currently, we're in the process of optimizing our product and planning for a clinical trial to validate our technology and go through the rest of the regulatory process to get this coating into market. Yeah, thank you for listening. And if you have any questions, I'll stick around until the end.
All right, thank you so much, Rafaela. That was very interesting. All right, so it looks like our next presenter is actually running a little late and having some problems logging on. So in that case, I would ask Greg if he's okay with presenting now. Otherwise, we can maybe stop and see if there are any specific questions for Rafaela. All right. Yeah, Greg, would you be okay with presenting a little bit earlier?
Yeah, no worries about that. Let me
Awesome.
just share the screen real quick. All right. Well, I appreciate the invitation to come share something that sits very dear to my heart and something that I've been putting a lot of my time and effort towards. So I'm an active MD PhD student at UC San Diego. I joined UC San Diego back in 2016 after graduating Duke with a undergrad degree in biomedical engineering. During my time at Duke, I also co-founded a separate company. It was a medical device company that is still ongoing. And so entrepreneurship has been a really important way, from my perspective, in order to take innovation within the lab and translate it to actual diagnostics or therapeutics that can help patients in the clinic. For me, it's unrewarding to simply have a career filled with lots of academic papers. I really want to see what I'm working on to ultimately impact the patient. And so I'm also here today presenting about a second company that I co-founded, Micronoma, in order to create new kinds of cancer diagnostics. And I think as you'll see amongst many of the startup presenters today, the science is important, and I think the stories are important too. And so I'm going to start with both of those. So the reason why I got involved in studying cancer at all is because during my freshman year of college, my grandmother, who's pictured here, who was otherwise a very healthy and active woman, suddenly was diagnosed with stage 4 pancreatic cancer. And by suddenly, I mean she had minimal symptoms, mostly abdominal bloating, some GI distress, some diarrhea, things that you would normally associate with a fatal cancer diagnosis. But as you can see here, pretty quickly after she was diagnosed, she started rapidly deteriorating. It was an event that shocked my family, not only from the diagnosis, but within a month, she actually passed away from the complications. I set out to better understand cancer biology as an undergrad student and wanted to go to med school in order to understand how pancreatic cancer was managed and treated. As I became more and more familiar with the literature, I saw a very disturbing trend, which is that since 1975, the 5-year survival rates for pancreatic cancer have improved less than 10%, despite massive, what we would call genomic revolutions in cancer. So even though our understanding of cancer as a disease of the human genome has evolved, our ability to translate that information into useful diagnostics and therapeutics for pancreatic cancer patients has not materialized. And so I started wondering, perhaps there's something that we're missing in this analysis. And during my second year of med school, I was astounded to see this really beautiful paper that came out in Science, which I'm showing you some figures here. On the left, you'll actually note that in 76% of pancreatic cancers that they examined, they were actually filled with not only bacteria, but chemotherapy degrading bacteria. What was also very astounding to me from this paper is that patients who underwent a very particular kind of diagnostic procedure to diagnose pancreatic cancer actually had higher amounts of chemo-degrading bacteria inside their cancers, meaning clinicians trying to do a good thing, which is diagnose your patient's cancer, were actually making those patients resistant to the very chemotherapy they were going to give them, which is a very bad thing. It actually violated the very, inherent principle of medicine, which is to do no harm. And made me start thinking about the abundance and prevalence of microbes in other cancer types. Fortunately, this wasn't the only paper that came out. A year later, this paper came out of MD Anderson. This was published in Cell, which showed that solely using microbial DNA inside of pancreatic cancers, they were able to stratify patient survival by 9 years. So you saw that the average 5-year survival rate was about 10%. So patients who survive up to 10 years are very, very rare. But for whatever reason, differences in the cancer microbiome were able to stratify patient survival. This was another paper that came out in Nature a year after that, demonstrating that it wasn't just bacteria, but in fact there were fungi residing within pancreatic cancer that were causing it to grow faster. So all of this made me start thinking, as I was getting ready to decide my PhD, why aren't oncologists talking more to microbiologists? And to do a brief review for those who are not microbiologists, most of life is microbial. If you took the history of the Earth and you put it on a 12-hour clock, microbes appeared around 1.5 hours within that 12-hour period. But Homo sapiens didn't appear until the last 2 seconds. If you look at all the genetic diversity on Earth, humans occupy a very small fraction of it. The vast majority of the genetic diversity on Earth is actually composed of microbes. And you can see here on the far right that microbes live in some of the most desolate environments that humans can never thrive in. If you count the number of cells on the human body, there's actually more microbes in it and on it than there are human cells. And if you count the number of unique genes, you'll see that there's only about 20,000 human genes, but there's close to 2 million unique microbial genes. So we're only about 1% human by gene count. And yet the supposed hallmarks of cancer, which are shown here, and this paper, along with its predecessor, has been cited over 84,000 times. So it's the gold standard of cancer research. There's nowhere here that describes microbial impacts on cancer, which is very surprising. And a small note on blood. We've actually known, and by we, I mean the scientific and medical community, that since 1977, there have been associations between transient bacteremias and patients who were later diagnosed with colon cancer. In fact, those who are familiar with biostatistics will note that they identified hazard ratios up to 17 in a meta-analysis of 13,000 patients in Hong Kong who had transient bacteremias and then were later diagnosed with colon cancer. So I devoted my entire PhD to try to answer this question. How prevalent are microbes across all major cancer types in the human body? And not only how prevalent are they, but how unique are they? And what we ended up discovering was something really fascinating, which is that cancers have tumor-specific microbiomes. So lung cancers actually have a distinct microbiome from colon cancers, which have a distinct microbiome from breast cancers, which have a distinct microbiome from bone cancers. What's really interesting about this is you can actually start building new kinds of diagnostics that entirely rely on microbial information to diagnose cancer. And that note that I'd said about blood, where transient bacteremias were later associated with the diagnosis of colon cancer, we actually identified small amounts of microbial DNA being leaked into the bloodstream of patients, and were able to not only diagnose the presence of cancer, but also the type of cancer. And we demonstrated this in over 20 cancer types in an analysis of over 10,000 patients and 18,000 samples. And this analysis was published in Nature last March. Now, as I'm showing some of the data here, this data was actually published in the Nature paper, but you can see that our performance compared to 2 other leaders in the field of liquid biopsy for cancer diagnostics is as good, if not better. What's incredible is that these other two companies were collectively acquired for $10 billion just a few months ago. And we've fortunately had the opportunity to raise about 6.5 million and are very excited to see that our performance is as good, if not better, than our competitors. But at the end of the day, this isn't just about trying to increase performance to be better than your competitors. It's about trying to save lives. And although we've been through a very terrible pandemic together, I will note that cancer is a problem that has been with us and will continue to be with us after the pandemic is over. There are 18 million new cancer cases worldwide each year. That number is supposed to go up to 21 million in the next 5 years. And perhaps what's most surprising of all is that there are 4 million preventable cancer deaths each year that could be saved with early detection. And so we've created a team. These are the co-founders, myself, Dr. Sandrine Miller-Montgomery, who used to be head of the Center for Microbiome Innovation at UCSD, who actually left her job to help be CEO of Micronoma, and my PI, Rob Knight, who is a co-inventor with me on Micronoma's technology. We've been joined by a fascinating scientific advisory board ranging around the world, from MD Anderson to the Weizmann in Israel, to UC San Diego, to Marty Blazer, who is very famous for studying Helicobacter pylori in stomach cancer. And so I would encourage you to, if you have an interest and you're graduating soon, to consider joining us in revealing new early stage cancers to save lives with microbial signatures. Thank you.
Thank you so much. That's an incredible story and really inspiring. Yeah, all your work with Micronoma is very interesting. So thank you so much, Greg. And Dr. Chen is now with us, so we will go ahead and move on to hydrostasis. So whenever you're ready, Dr. Chen, take it away.
Hi, everyone. Let's see if I can share these slides. So I took a little bit of a different approach to presenting my presentation to you guys today, because I wanted to highlight the fact that you really don't have to know what you want to do to eventually end up somewhere where you start your own company. So I really call this career spaghetti. So just to give you a little bit of a background, I was born in Taiwan, and I did bioengineering at UCSD, worked in several undergrad labs. I was mainly interested in imaging. And at the time after I graduated, I didn't quite know where I was going. I applied for bioengineering PhD programs because I was really searching for interesting projects. And that's really what I was going for. And to be honest, I was rejected to a lot of programs. And I ended up in this program at Tufts University. And it sort of was it was interesting because I started doing these high risk, high reward projects, which is what I have ended up doing all of my career. There are certain projects that are well funded, millions of dollars going into these large projects, but I ended up doing sort of like a side project because I thought, okay, nobody's ever done this before. Let me try it. And so I ended up doing my PhD on developing a diagnostic tool for early detection of peripheral neuropathy. And then I said, okay, well, Boston's too cold. I want to come back to San Diego. What can I do to continue my learning? And I think that's really the thing that drives me, is I'm a lifelong learner. And again, to bring this back up, I didn't really have a clear idea of what I wanted to do. And so I just followed my instincts of, okay, well, I did an entire PhD on, diagnostic tool for peripheral neuropathy, but as a biological mechanism, I didn't actually quite understand what the biology was behind it. And so I ended up doing a postdoc at UCSD pathology and really looking into the biological methods or the biological process of peripheral neuropathy, specifically corneal confocal microscopy. Ended up developing a optical model and some hardware to be able to look at animal models in corneal confocal microscopy. So then, and then I said, well, what am I supposed to do now? You know, either I go and apply for professor jobs, and this was during the time, I think it was very difficult to find jobs during that time. So I said, you know what, I'll just continue with my schooling. And I did another postdoc, which, by the way, I don't encourage everybody to do so many postdocs. I actually, now looking back at it, I could have had various different careers. But again, this is what I'm talking about, career spaghetti. You don't actually need to know what you know, are eventually going to do. So I went into another postdoc at Scripps Oceanography, really because I wanted to develop more technologies. And this time I developed a 3D scanning microscope, or was on the project to develop this microscope for real-time phytoplankton identification. And this was a scanning microscope. And, you know, that was also very fun. And then I learned how to handle big data, because it was terabytes of data coming in, and how do you segment, how do you find the interesting data, how do you store the data in real time? And that really led me to Sanford Burnham, which is in Torrey Pines. And I started managing the microscopy core there. So all of my sort of positions or jobs, if you will, has this sort of thread of imaging, of optical imaging. And I started going into drug discovery. And a few years after that, I decided to start my own company, which again has nothing to do with any of my 3 other posts, right? So I think the moral of the story here that I wanted to sort of impress is that you don't have to know exactly what you're doing. I followed, you know, interesting projects, which eventually gave me skill sets of, you know, how to sort of use all of my skills. And I'll talk a little bit about the skill sets that were not used in my previous jobs that I really think are really great for starting your own business. And these are the soft skills. So, you know, as engineers, you know, so my PhD was about engineering. And so as engineers, your soft skills are really not appreciated as much. I realized I was really great at relationship building, right? You have to care about the other person, you're about team building. I was very empathic, where, you know, I you know, really tried to communicate with my customers. I really try to communicate with my teammates, collaborators, etc. I used a lot of transparency and to communicate well, leadership, and all of these things are really soft skills that you need to also hone. If you're missing these skills, you need to hone if you want to go into you know, a startup or starting your own startup, because really it's about building relationships and being able to sort of sell your idea. But I realized that because these were underappreciated in my you know, engineering background, this is where I excelled. So now I have this engineering background and I had the soft skills to be able to go into business as well. So I think I didn't quite appreciate these you know, other skill sets going into it. So I just wanted to sort of mention that as well. A really quick, you know, startup pitch frame, if you will. If you know, have been watching pitches or Shark Tank or whatever, it's always why you, why this, and why now, right? And so just thinking about all of these things, like why you, right? What are your, you know, unique background that is going to make you successful in doing this particular, you know, project, you know, invention, if you will. or what, and why this? Why are you solving this problem? Is this a problem that's going to make money, right? Because a lot of times as scientists, we go in to solve a problem that we think is very impactful, but you have to understand that this is a business, right? How are you going to make money? And if you get investors involved, those investors want their money back times 10, right? And so you have to be understanding of all of those things. Again, this whole team building, collaboration, empathy thing comes into play, because if you're going to get investors on, you have to think how they think. If you're going to get partners on, you have to think how this benefits the customer. If you're going to hire employees, how are your employees feeling as you're leading this team, right? And so all of these things that I think was not clear to me when I started off, which I think is important to highlight. And of course, why now? I think, I forget what the title of the TED Talk is, but it's basically like why startups succeed. It's not about the team. It's not about the technology. It's not about all of these things that they studied. It really is timing. It comes down to timing. Are you, making a painkiller? Or are you making a vitamin, right? Because when you have a recession, for example, people can just not buy that vitamin, right? But they will have to buy the painkiller because it really serves to solve this problem that they have. Oops, I don't know why it looks funny like that, but so my origin for hydrostasis is basically turning my personal pain into passion. And that really is from my background in sports as well. So I'm actually, used to be, at least before the pandemic, an amateur kickboxer. And so if you know about competitive sport, competitive, martial arts, there's weight classes, right? So you have to be a certain weight, and you have to, a lot of times, dehydrate yourself, which is what like a lot of people do, and it's very dangerous. At the same time, I've noticed there's a lot of health implications that is not addressed for hydration. So just for example, it's hard to see on the bottom here, 1 to 2% of dehydration is really when your thirst begins. That's really when your body's already telling you you're already dehydrated, right? And then there's significant cognitive decline there as well. When you get to 4 or 5%, you have headache, irritability, sleepiness. This also implicates a lot of mental health as well. So if you have brain fogs or if you're just moody, sometimes that's because you're dehydrated. And it's so easily controlled, right? And why don't we have something to tell us when and how much to drink? And of course, when you get to, further down the line, you have heat stroke, respiratory problems, renal failure, and of course, death that you hear every single summer. This is San Diego. There's, kids on the turf that, faint, because of, heat stroke and dehydration. So these are all very preventable things. And so if we can have a device that can, solve for these things and really give you preventative information, that's really what my purpose was. Just a little bit more on, again, the problem, right, of the startup that we're solving. 3 out of 4 adults in the US are chronically dehydrated. 3% water loss can lead up to 35% loss of muscle endurance. There's 10 million adults over the age of 65 that are admitted to the hospital for dehydration every year. And if you properly maintain hydration at home for these older adults, you can save the hospital system, the US hospital system, $20 billion. So this goes into remote patient monitoring. So I hope you can see how I build that story, right? This is my startup pitch. These are your problems that you're trying to solve, and what is the money that you're going to make, right? This is sort of the market that you can, impact. Again, there's no alternative, right? So everybody's asking, okay, you're doing a business, you're doing a startup. What are your competitors, right? So here's my pitch, right? There's no good alternative. Your sense of thirst changes with age, it's too late. There's bioimpedance measures that are inaccurate. They're very motion sensitive. There's rules of thumbs, right? There's apps that you tell it how much water you're drinking, which is not really actionable and not personalized. And then urine color, of course, is pretty crude. And then urine specific gravity is also delayed. You have to pee in a cup. You have training to use the urine specific gravity as well. So that is, you know, why there needs to be an improvement on the technology. And then this is my timing, right? So I'm saying that, hey, the performance sports have been trying to solve this for years. You have consumer wearables focused on health. Apple and Google are investing billions of dollars in their next health frontier. The device manufacturers are looking for the next big insight. Hydration is really on everyone's roadmap. This is from our partners. And then remote patient monitoring, of course, is taking off. It was taking off before COVID. Now, of course, it's you know, exploded. And then this is what we offer. We're offering a plug and play solution for device manufacturers. And this actually was a couple pivots. We started off making our own wearables, and we realized quickly that we're going to be in direct competition with the Apples and the Samsungs and the Garmins and Fitbits. And so as a business decision, we said, you know what, nobody wants to wear, you know, a watch and a chest strap and another wearable and this and that. And we were singularly focused on hydration. So what we decided to do was, okay, so let's make this a solution for the devices. So now we're selling to smart device manufacturers, and we provide what we call the 3 Ps: personalization, precision, and performance. We have a guided calibration, which is your personal hydration index. We're aiming for clinical grade accuracy. We're outperforming urine specific gravity. And of course, we give you this actionable information, which is fluid intake, guidance, benchmarks, gamification, and data sharing. We also have a nice IP portfolio as well to sort of obviously protect our IP. So we're able to detect increases in hydration after drinking, decreases in hydration after exercise, and we're 30% more accurate than urine specific gravity. We can confidently detect both loss and intake in 0.5% of the body water. This is actually before thirst. And we are successfully predicting hydration versus serious dehydration states in 100% of our test cases. So now we've done sort of the groundwork internally, and then we're going out to our external testers to test, you know, more elite athletes, more older adults, people of different color, skin, all of these things that, you know, impact biological signals when you're using optical technologies. So the idea basically is to be able to give you this optimal zone, right? So you have overhydration, which impacts patients with kidney disease or heart failure. And then you have dehydration, obviously, for the general population, diabetics, as well as elite athletes. And then we're using 4 different indications to give us our proprietary algorithm: skin tone, blood flow, fat content, as well as hydration. So this is just a little summary of what you need to consider, right, for commercial success for a wearable health tech. You have to do lots of research and development, obviously, and make sure that your data is relevant and obviously get to the clinical accuracy that you want, or not. You can go towards the consumer tech as well. Form factor is important, right? You don't want it too big or too small, or, you know, you want to make sure that people can actually wear it. Actionable data is really what's important these days. It's like, you can give so much data as scientists. You're like, yeah, I want to give you all the data, but unless somebody knows how to use it to change a behavior, it's not usable. It's not interesting. And in fact, people like hate data overload because we have so much information out there. Compliance is a big deal, obviously, in the clinical sense, but also just like UI UX, right? How does the person use your device? And personalized and preventative is really what most of the health tech, at least in the wearable world, is going towards. How do we prevent early onset of chronic diseases like diabetes, like Alzheimer's, like kidney disease? You know, how do we prevent that, and how do we predict that, and how do we avoid that if we take care of ourselves earlier? And of course, lastly, and really not lastly, it should be firstly, is how much does these things cost? Right? Who exactly is your customer, right? So for example, if you're selling to the hospital system, you're not actually selling to physicians, right? You're actually selling to the payers, right? And then you have to think about reimbursement, you know, who's actually paying for this? What are the CPT codes that you're using, regulatory stuff. So all of these things, like when we go in, at least for me, when I hopped into the startup world, I'm like, okay, great, I'm gonna develop this technology, right? But there's so many things to consider to be a successful business. And, you know, I'm early on in my career as an entrepreneur. We only incorporated in 2018, so I'm still learning. I'm sure I'll be learning for quite a while. But you know what, like, I think I'm the happiest I've ever been as far as career goes, busy as I've ever been. But I think the reason why is because it really melds all of my soft skills and the science background as well. So, you know, I encourage anybody that's interested to make that jump. Obviously reach out to me. You know, I think perhaps Rachel has my, you know, information that, you know, you can contact her. But yeah, I'm happy to answer any questions. I've been mentoring quite a bit as well in the local accelerator programs. I've been a speaker at the Basement. And last year I was also a entrepreneur in residence at UCSD as well. So very happy to help talk about any ideas and, you know, any questions that you guys might have. And that's it.
Thank you so much, Dr. Chen. It was really nice to hear kind of the other side, not just the science, but everything else that's involved. Awesome, thank you so much. All right, so our final presenter before we open it up to questions will be Dr. Carson from PensaVision. So Dr. Carson, whenever you're ready, feel free to take it away.
Thank you again for giving me a chance to speak. And talking today about my company, PensaVision, that I founded with a colleague, a UCSD professor, and where we're a medical device company, and we do 3D imaging at low costs. So to give you an idea of some of these things that we're talking about, so everything comes into focus, and when we talk about low cost, we're talking about something like our entire device might be a couple $100s, and that includes the computer system. We run our device off a Raspberry Pi computer, all of our hardware, all of our optical components. I think our most expensive one is maybe $45. Also, we're doing 3D imaging at low cost, and we're doing it with a single lens or single perspective. And just to try to, you know, explain why that's important, all of us can do depth perception by having two eyes. You know, you can go to a 3D movie where they use a whole range of cameras to maybe get 3D, but here we're doing 3D with a single perspective, as opposed to what your eyes do, which are stereoscopic imaging, where you have multiple perspectives. And the fact that we do 3D imaging from a single perspective also means that we can go down to very, very small scales where we're not dependent on having a big distance between, you know, two perspectives. We can go down to microscopic scales, the kind that you might see in an endoscope, and we can still get high resolution imaging. And part of this low cost, and it's also meant to be something that's very easy to implement in a resource limited setting. And so that might include, for example, sub-Saharan Africa, maybe in a rural village that doesn't have electricity. That would be, this device should operate, you know, it should operate very well there. Since it's battery powered, it's portable, it's handheld. And this is optical imaging we're doing. So we're not doing subsurface imaging, we're doing high resolution imaging of a surface. Okay. And this is, of course, Translational Medicine Day. And I think PensaVision certainly was built on the spirit of translational medicine, of taking ideas from one field and passing it to another. So to give you an example of that, my background is, it's not in medical devices, it's not in bioengineering, it's not in engineering or medicine at all. I've spent my entire career in astronomy, doing searches for extrasolar planets using things like the Hubble Space Telescope, and using very large telescopes on the ground to look for planets around stars. And the idea behind PensaVision is we took a lot of these tools that have been used at places like NASA or used in astronomy imaging, and applying those to inside the body. And I'll talk a little bit more about that later. The crux of what we're doing, or why what we're doing offers a lot of potential, is that it really comes down to the fact that anatomy is three-dimensional and a typical image is two-dimensional, and kind of that fundamental disconnect between a two-dimensional image and three-dimensional anatomy is really the reason why three-dimensional measurements have had a huge amount of success across a wide range of field when it comes to diagnostics and screenings for all sorts of types of cancers and growths and other types of diagnostics. And so that's the idea behind what we're doing. If we can get high resolution 3D images and do that with simple materials and then make it easy to implement. And the approach we're doing, I mean, it can be applied to anything. It doesn't have to be applied to medicine. But our main application that we've been focusing on is cervical cancer, doing screenings for cervical cancer. And cervical cancer, I mean, to give you an idea, is the #1 cancer killer among women in Africa. And there's a number of different ways, I mean, as I'm sure you're aware, preventing cervical cancer. But for a variety of reasons, many people don't have access to those things, or they do not wish to use those things, including things like HPV vaccinations or regular screening. So part of what we're doing is giving a way to do a screening in one single visit. So for example, if that patient might visit a healthcare provider, maybe if they visit a healthcare provider once every 10 years, during that visit, we can do, the idea is to do that screening and to be able to decide if an intervention needs to be made, and to do it with high resolution 3D imaging. So it's complementary to all of the really critical preventative methods or preventative tools for cervical cancer. It's certainly not in competition with something like an HPV vaccine. And part of it comes down to the idea that with doing 3D imaging, you don't need labs, you don't need to send it off anywhere. So it's very telemedicine friendly as well. You take the image right there, and as long as you have, the equivalent of a cell phone connection, you should be able to make a decision, or at the very least triage, and figure out, which are the 99% of patients that are maybe low risk, and who's the 1% that you do want to get them to a specialist, or do want to get them to a hospital. So here's an example image. This was of a cervix we imaged that had recently been removed via hysterectomy that we did at UCSD. And a lot of what we're doing here, so we're generating high resolution 3D images out of a series of two-dimensional images. And, our journey was, if you look at this sort of, those 3 images there, we start off with a two-dimensional image, and it took me about, I don't know, 4 or 5 months to figure out how to get a kind of poor quality 3D image, which is that thing in the middle that you're seeing, where you sort of get the basic shape of what you're seeing. And then it took about 4 or 5 years to develop all of the artificial intelligence and machine learning to basically take what's a really, really messy 3-dimensional image and extract from it this beautiful high precision 3D image that you're seeing on the right, where it's high quality enough that you can save someone's life, because you can, by being able to explore and be able to screen for things like precancerous growths. And our serve image device, as you're seeing up there in that cartoon, is just a simple handheld device with a very slender tube that's meant to be inserted in the vagina for imaging the cervix. And to give you another example of that, this is just something, was observed of a mole on one of our engineer skin to give you another example of the kind of 3D images that we get from this. The technique we're using, I mean, again, it's easy to do poorly. It took us a long time to develop all of the artificial intelligence and machine learning to extract from what begins as something like very, very messy, to extract the true information from the information that's false positives and noise and all those different sources. So I mentioned how PensaVision forms. So I spent my career, most of my career, doing searches for extrasolar planets. So like this image you're seeing on the bottom right there. So that was a discovery image by my students and I a few years back of an extrasolar planet. And the reason why some of this is applicable, so an image like this, you might hear about or see an astronomy image, and you look at it and it looks like someone just took a photograph and snapped a photo. This discovery image on the right, I mean, we took that image at the telescope. We started off with like a stack of 150 images, and it took maybe 6 months of basically manipulating that stack of images to get to this final image here, using a lot of these AI tools. And part of that was using digital mask to like digitally block out the starlight and see this faint planet nearby. And so we took that principle of using a lot of these tools in astronomy and applying it to looking inside the body. And it started off from a conversation that I had with a professor of medicine at UCSD, where we talked about maybe there was an opportunity to be able to improve imaging techniques and in some of these screenings that we're doing, and using this approach. And I should also mention it's not only some of the imaging things. So, you're looking at outer space, you see something that's blurry, it's faint, it's noisy, it's hard to see. You have to make sense of it. You have to figure out what it is you care about, what it is you don't care about in the image, which is the same as looking in the body. It's blurry, it's messy, it might be changing over time, the same way looking through our atmosphere, things are changing over time. The approach we're doing, we also borrow a lot of our telemedicine approaches from astronomy imaging. So to give you an example, we're trying to figure out how to have a device that's in sub-Saharan Africa. It can communicate with a doctor at UCSD over the equivalent of a poor cell phone connection. And so you might wonder, how do you transmit a high resolution 3D image over a poor cell phone connection? So to give you a counterexample, you might have heard about NASA landed a new rover on Mars this past week. So that rover on Mars, it has to be operated and send back data over the equivalent of like roughly around 100 kilobyte per second connection. And I'm guessing most of you guys were not alive when Wi-Fi was that poor of 100 kilobyte per second. I mean, it's worse than a poor cell phone connection. And so if NASA can operate a $3 billion rover over 100 kilobyte per second, we can communicate a high resolution 3D image of the cervix or of inside the body and get it to a doctor at UCSD. And we use it's very similar strategies, actually. So I'll just say briefly to wrap some things up here. We recently submitted an NIH grant to do patient testing, and we got back the results. And in a very rare instance, they gave us a perfect score on our application and approved us for funding. And I actually thought it was a mistake because in, 20 years of writing grants, I've never got a perfect score on anything. But I think it reflects how the NIH has been very enthused about what we're doing, and shows that there does seem to be something special about what we're doing. And we've been able to gather that enthusiasm. And so just to finish up, looking just close to the future. So we have a 20 patient pilot study that should be starting hopefully in the next month or two at UCSD. That's led by Dr. Jessica Kingston is going to be leading that study. It's the first time we're getting patient data. And our upcoming is trying to get working on FDA approval, and also being able to get 3D imaging, but using a less than 2 millimeter diameter endoscope. Okay, I'll stop here. I know I'm about out of time. Thank you for giving me a chance to share my journey.
Great. Thank you so much, Dr. Carson, and thank you to all of the presenters for your wonderful insights. So we do have some prepared questions. I don't think we'll have time to go through all of them, but I can just start off with one prepared question. And if anyone has to run once it gets to 5 o'clock, totally understand. But I guess just the biggest question of all is, what are some of the pros and maybe cons of working at a startup compared to maybe a larger company, if any of you have that experience or know from outside perspectives?
I can start. If you are a student and you want to work at a startup, you're going to learn a lot. You're going to do all kinds of things that perhaps were not in your job description, or maybe if it was a good job description, it would just kind of be, you are all hats, everything. And this is for early stage startup, maybe like a seed stage or even Series A. You're just sort of like, you know, high fast paced, doing whatever it is, you know, at the moment that needs to get done. For a sort of larger corporate role, it's a little bit more stable, right? The pace is sometimes less fast, sometimes still very fast. It depends on obviously the groups that you're working with and the projects, of course. But you're much more sort of structurally contained, I would say. And your job description is much more narrow. So, you know, if you're into a little bit more chaos, a little bit more fast paced, a little bit more uncertainty, I think people have to be very okay with that. Then I think startups are a great way to learn, and you learn a lot of different skill sets, not just, you know, the science of it. You sometimes, you know, get pulled into marketing randomly, right? And you're like, I don't know anything about marketing. But that's great, because any other job after that, you're going to have, you know, skill sets in every you know, field.
Does anyone else want to hop in on that?
I totally think that idea, I mean, like Dr. Chen was saying, that you have to, if you're not an expert at everything, you have to be an expert at everything. At least you have to do everything. So I know people in my company, the engineers, they give me these wild looks because I tell them, okay, you need to be our expert on material science, you know, on nanograde polymers. And they say, but I'm a computer programmer. And I say, I don't care. Like, we can't afford to hire a materials physicist. So you are now our materials physicist. It doesn't matter. Your background is in something else. We need you to do this. And that sometimes can be really shocking to people to hear all the things they're responsible for doing.
I can add a little bit of a drawback. I think, you know, one is the uncertainty of a startup. Obviously, you're talking with a panel of biased people. We're all doing, you know, we've all gone to the startup side, right? But one of the drawbacks is that, you know, depending on the funding situation, right, you might be pulled in this project or that project, or perhaps the startup might just, you know, die because they can't get funding or whatever, right? Like if your startup is operating under an NIH grant and that funding, you know, is stopped, and then you have another grant, now you have to work on that project. Or at any point in time, if the market says so, that the business model has changed, or your customer targets have also changed, then now you have to be okay with that as well. So I think just like very fast-paced, high risk, but also very high reward, and being okay with, you know, sort of a chaotic world is where the startup is.
I think you have more responsibility at a startup company. I think everybody has more responsibility because, at least for us, like we can't afford to hire someone who's just an assistant. they have to be in charge of something. They have to be like your, head manager in charge of this. It can't just be like, oh, you're there to assist somebody else in doing a task. No, you have to be in charge of that task. You have to take the initiative. You have to potentially hire people below you, even though you might not feel like you're necessarily, ready to be in that position of high responsibility. But it's just the requirements of it.
All right, well, we are 2 minutes past 5, and I want to be respectful of everyone's time, so we'll wrap it up here. But thank you again to our 4 presenters. We're so honored to hear from you, and your stories are incredibly inspiring, and we wish you the best of luck with your future endeavors. And yeah, so that will conclude our startups panel. Next up in TMD, to wrap it up, we have a networking reception. So you all should have gotten the link emailed to you. It'll happen on gather.town. And yeah, we'll see you there if you can make it. Bye, everyone. Thank you so much.
Thank you. Thanks, Rachel. Thanks, everyone.
Thank you.
Thank you.
Bye.
Bye.